Good day, everybody. In this lesson, we're going to be looking at a specific group of proteins called enzymes. If there was ever a beautiful example of how structure relates to function, you'd have to look at enzymes. Enzymes are responsible for controlling the chemical reactions that are taking place inside our bodies. So we'll be looking a lot about enzymes as we study digestion, cell respiration, and just about every single process that takes place within the human body. Now, affecting reaction rates, there are several things that will control how fast a reaction proceeds. All right, among these, first of all, is the concentration of the reactants. In order for a reaction to occur between chemicals, those two molecules have to contact each other. They have to come in contact somehow. Now, by this first bullet here, the concentration of reactants, if you have more reactants, there's a greater chance that the two reactants that are involved are going to come in contact. The more chances the more times they react, the faster the reaction goes. So, by the reverse, if there's very few of those reactants, the reaction goes very slowly. Second thing is the temperature. If you look back at all of the old science films here, the mad scientists are sitting there at their desks with a beaker of boiling water or whatever. We need to increase the temperature to increase the reaction rate. By increasing the temperature, you're increasing the molecular movement of the reactants. As the reactants are moving faster, again, you're going to increase the number of collisions, and the reaction will proceed at a faster rate. Third thing is the presence of enzymes or catalysts. In your chemistry class, you learned about catalysts, which are substances that will increase the rate of a chemical reaction without themselves being consumed. Well, enzymes are organic catalysts. They're going to speed up a reaction without becoming involved. And this is going to be the main point of our lab that will be following up after this lecture. As I said before, an enzyme is an organic catalyst. Organic means that it comes from living things. And as such, they're going to have special properties that we'll be investigating in lab. Now, enzymes work by lowering the activation energy of a reaction. If you look at my chart over here, this black line shows the rate of a reaction without an enzyme. Now, this is an energy barrier, what we call E sub A, activation energy, without an enzyme. Energy of activation without the enzyme is this line right through here. Every substance that we have in the world is going to have an energy of activation. For example, sugar. If you let sugar sit out on the table long enough, maybe a couple thousand years, it will break down into carbon and water and the constituent elements of sugar. But because we have this energy barrier, the sugar molecules have to overcome this energy barrier before they can actually break down completely to water and carbon dioxide and water. Enzymes, in the presence of an enzyme, the reactants require less energy. So the reaction proceeds at a much faster rate. Enzymes work, and the best way to describe this is what we call the lock and key model. There's an enzyme substrate specificity. In my picture over here, this down here is the enzyme. Now, enzymes are proteins, and they're configured in a specific way. This enzyme will only work on this substrate. The substrate has a particular shape that will slide right into the active site on the enzyme, and the enzyme will act on that substrate. It might break it apart. It might join two things together. Now, to help enzymes do their job, there are cofactors and coenzymes. A cofactor is an inorganic substance. that will bind to an enzyme and make the enzyme alter its shape so it'll work with a substrate. You know, a lot of cofactors would be like inorganic molecules. Coenzymes are organic, so it might be like vitamins that will bind to the enzyme and actually allow it to perform its function. Now, when we start talking about naming enzymes, most of the enzymes we're going to be working with are going to always end in ASE. Like with carbohydrates, I always said a sugar could end in OSE. Well, this is another way to recognize the name of an enzyme. So even without knowing specifically, that it is an enzyme, if it ends in ASE, you have a pretty good chance that that substance is going to be an enzyme.